So it's a great pleasure to have Karthik Shekhar here today to kick off our seminar series for the new year. Currently a professor at the University of California, Berkeley. Prior to that, he was at the Broad doing a postdoc, and we're really excited to learn more about bioelectrical dynamic membrane interfaces.
Thank you. It's a pleasure to be here, and thank you for the invitation and its space. It's always nice to visit Chicago at this time. It brings me back to the memories of when I was in Boston. Certainly not missing the code. Okay, so today I'm going to talk about a project that has been fairly recent, and I would say has not been the main focus of my group for a while, and so it's interesting and also exciting to present it to an audience. So my group works at the intersection of neuroscience, computation, and biophysics, and we've generally been interested in two kinds of problems. So on the one side, we have been interested in understanding the molecular underpinnings of neural diversity and relating that to structure and function. And much more recently, we've become interested in this question of neuronal electricity or bioelectricity, and that's sort of what I'm going to talk about from the perspective of looking at charge dynamics at membrane interfaces and what it could mean for signals propagating along these membranes. And of course, we are not the first to do this or to think about this problem. And so as I go along, I will certainly acknowledge the works that have inspired our thinking on this. And so to acknowledge, this is a close collaboration with my friend and colleague, Kranthi Mandadapu, who's a continuum mechanician and a physicist. And the work that I'm going to describe today was led by a postdoc, Hyungju Roh, who was actually in the job market, and a grad student, Josh Fernandez. And over the last couple of years, we've also recruited a couple of others. So for example, Sirui Ning is a recovering string theorist who's joined the fold. And Jafar has also been working on some problems. And so what I want to motivate this is that ultimately the electrical signaling is nature's solution to robust, rapid information transfer. And so there are a few videos here, quite fun. So you can think of human reaction times. It's quite impressive, of the order of 200 milliseconds. And if you think that's impressive, let's look at a cat. And cat has an order of magnitude lower reaction time as it's slapping the snake. And then this is probably the most, from my understanding, this is one of the fastest reaction times in biology. It's the mantis shrimp, which is this little creature that lives underwater. And it has these two appendages that are its punching machines. And it's able to strike with such a force that it actually produces bubbles and cavitation in the deep sea. And as you can see, a crab that tries to mess with it, it does not end too well for that crab. So it's made a punch. And there you go. So, and so all this means that signals propagating along the body of the organism have to be really fast. And so electricity really beats any other mode of signal transfer, certainly hydraulics or mechanical waves and also chemical waves, and definitely orders of magnitude faster than how a diffusive signal could sort of traverse that distance. And of course, all of this is very well known historically, as starting with the works of Galvani and Volta, all the way to Hodgkin and Huxley. They studied the action potential. And the action potential, for those of you who may not know, is basically a transient change in the transmembrane potential of a neuron that propagates along the length of the axon. And conduction speeds have been measured right from the time of von Helmholtz, and they go anywhere from a meter per second to 200 meters per second. And this is an ionic phenomena. It involves sodium, potassium, and various anions, but primarily sodium and potassium. And the major, I would say, theoretical work or theoretical model of how the action potential works stems from the pioneering work of Hodgkin and Huxley. And what they really did experimentally was to use as a model the giant axon of this squid. And what's interesting about the giant axon is that it's giant, and it's big enough that you can stick an internal electrode. And so they were the first to actually measure the transmembrane potential. and show that during activity, the transmembrane potential changes from being negative to positive. And then the big question sort of was, what's the underlying mechanism of this? And so Hodgkin and Huxley measured this in Woods Hole, and then they went out to fight the war. And then once they came back, they solved the problem. And so this is a much more modern measurement of the action potential using a voltage sensor from Adam Cohen's group. And you can clearly see here that there is a sort of a wave that of transmembrane potential change that traverses the neuron. And so what's the Hodgkin-Huxley model? So it's essentially a circuit model. They assume that the neuronal membrane is a circuit, and so it acts like a capacitor. And what they also assumed is that every type of ion can permeate the membrane and has its own conductance, and every ion has its independent conductance. So once you make these assumptions, you can write down from Kirchhoff's law a circuit equation, right? And their genius, in some sense, was through a series of careful experiments to figure out that the conductances for potassium and sodium were actually voltage dependent. And so depending on what the transmembrane potential is, the conductance of sodium can be higher or lower, and potassium can be higher or lower. And once they were able to fit their data, they were able to reproduce the essential phenomenology of the action potential. And so, of course, the big question then was, how can a membrane alter its conductance so acutely to different species? And of course, they didn't know the answer. They had a few different models, some of which were wrong, some of which were reasonable. But the solution turned out to be ion channels, which were discovered a few years later, hypothesized by Hiller and others, and then structurally characterized, starting with the work of Roderick McKinnon and several Nobel Prizes. So there are voltage-gated ion channels, all of which open and close in response to membrane voltage. And much more recently, the excitement is on mechanically gated ion channels or tension-gated ion channels. By the way, please stop me if there are any questions. All of this is background, so hopefully this is clear. And so the question that we became interested in, so of course there are many, many different types of ion channels. They've been characterized. There are voltage-gated, mechanosensitive, ligand-gated, pressure-gated. And what's also interesting is that the way these channels are distributed in different parts of the nervous system is not random. I would say the extent of super resolution data that we have of channel composition is still on the lower side, but this is a beautiful image from Kerju while working with Jiawei Huang, shows that in this axon initial segment, there is a beautiful lattice structure of sodium channels that sort of alternates with the spectrin rings. And so there is an intimate coupling between voltage sensitive and also the mechanics of the membrane. And so we got interested in this seemingly simple question, which is what happens when a single ion channel opens? So what I'm going to talk about for mostly half of my talk is, let's say I have an ion channel that is opening, and so that's going to be a flux of ions through the membrane. And it's a spatially localized flux. And what I'm interested in is sort of what happens as a function of space and time. And the way we were thinking about this problem is, how do ion channels potentially communicate? Right? So if the opening of an ion channel or a current produces a perturbation in voltage or mechanics, how does that propagate along the membrane? That's the question. Now, of course, initially we were thinking, has people thought about this? And of course, people have thought about diffuse charge dynamics. But then if you look on the web, there are various videos, and this is one such medical animation video of how the action potential propagates. And so you can see that, there's a flash of light that's sort of going around. Ion channels are opening and closing. And of course, this is an animation. And so there's no understanding of what are the time scales involved, the length scales involved. And so the other point is that also the coarse-grained Hodgkin-Huxley model and its cable model derivatives don't have explicit ion channels. And so we were interested in just understanding what happens when a single ion channel opens. And so the questions that sort of guide this work is sort of, how do these macroscopic features of voltage emerge from these microscopic elements? How might channels communicate with each other? And eventually, we also want to think about the role of membrane geometry and mechanics. And so why is this? It's seemingly a very, very simple question, but this is what makes it hard. So think of the channel being not an ion channel, but a channel for a solute, like glucose or something else that is not charged. And so there, if there's a flux, clearly we know that there's an excess concentration here, there's a reduced concentration here, and as a function of space and time, we expect it to be purely governed by diffusive dynamics. Okay, so that's a spherical sort of symmetry. But what happens when you have an ion channel? So when you have an ion channel, let's say you have a cationic transporter or cationic channel, when there's a flux, there's an excess concentration here, reduced concentration here, the same way, but then there's also excess positive charge and excess negative charge. And you have to globally, you have to be electroneutral. You're not allowed to break that. And so as a consequence, you are not going to expect things to just diffuse away like they do for solutes. So you would expect naively that the two have to attract each other electrostatically, which means that they have to somehow stick to the membrane. But the question that I'm interested in is how does it then follow in plane? How does it spread in plane? Is that a diffusive process? And is it just that they're going to stick like a pancake? And so that was the question. So that's the question that I'm going to think about for the next few slides. And the mechanism and the speed and so on. So the equations that we're using have been known for over a century. So there's nothing fancy here. These are the Nernst-Planck equations. Okay, so these are concentration balances. And for simplicity, we are thinking of a binary system that is monovalent. But and that's because we're also going to do some analytics. But in principle, the same equations would apply if you have a multi-component system with multivalences and so on. So just for simplicity, I'm going to think of the universe being comprised of a positive ion and a negative ion. So we have mass balance, just says that ions cannot be created or destroyed. And then this is the flux of ions, how they migrate. They migrate through Fickian diffusion. They also migrate down the electric field. That's electromigration. And then finally, to close this problem, you have to satisfy Gauss's law, which says that the electric field has to be related to the charge density, the free charge density. And so a few things here, which is that, again, we are not the first to apply this to biological systems. There's a rich history. So these equations were formulated by Nernst and Planck. And then there have been a large number of proponents in different areas. So for example, the earliest work on electrolytes by Debye, Huckel, and Onsager use these equations. They were applied to semiconductors in the 1950s. And then to acknowledge people in this neighborhood, Bob Eisenberg applied it to ion channels and definitely recognized the role of permanent charge in ionic transport, electrohydrodynamics, Melcher, Taylor, instabilities, electrochemical systems, Newman from Berkeley, Martin Bazant at MIT, and so on. And then, of course, they've also been applied to membranes and membrane stability with Professor Vlahowska from Northwestern being one of the much more recent pioneers. Okay, so these are the equations that we're going to solve. But even in the simple problem, the reason it's kind of complicated is because there are length and time scales that span almost 5 orders in magnitude. So what are the length scales? So we have the pore opening. And of course, I want to just be clear that we are not solving the Nernst-Planck equations inside the membrane. They're all only being solved in the bulk. But nevertheless, there's going to be a flux of ions, and that's going to be a length scale of 5 angstroms. And then you have the Debye length. The Debye length naturally arises because this is the length scale over which electroneutrality is broken. For biological systems, it's of the order of 1 nanometer. There's the membrane thickness, 4 nanometers. And we're solving it for system sizes up to 1 millimeter, and I'll tell you why we're going there. So that's already 5 orders of magnitude. Time scales, we have the smallest time scale is the so-called Debye time, which is the time for diffusive reorganization in this Debye layer. That's around 1 nanosecond. And then open times for channels can be of the order of 1 millisecond. And so what we're doing here is we're solving the full Poisson-Nernst-Planck equations numerically using the finite element method. And that is done because the inherent PNP equations are nonlinear. I also want to note that we are neglecting things like surface charge or permanent charge, which certainly Bob has written a set of beautiful papers to emphasize their importance. But under a lot of circumstances, at least in the bulk, as I will show, these equations become linear. So all those elements that we are neglecting are can be superposed on these solutions. And of course, just want to be clear that we are not looking at transport within the ion channel. Okay, so the numerics are going to be solved by the finite element method. And for this problem, we had to sort of work carefully to design conformal meshes to make sure these things are convergent, do a lot of error analysis because the initial solutions were, let's just say, a little surprising. But then now we've gotten good at it. And I will also show that all the numerical solutions are also substantiated by analytical formulae. Okay, so let's look at two variables. So for a binary system, you can do this nice transformation where you can define two auxiliary variables. So you can define the overall salt concentration, which is just the sum of the concentration of the cation and the anion. And then conversely, you can define the difference, which is the charge density. So instead of C plus C minus, I have C and rho. And so let's look at these two variables. So these are heat maps talking about the temporal evolution. So by the way, the system is, we are using cylindrical coordinates because we are assuming axis symmetry around the channel. So you have R and Z and time. And so these are heat maps showing the evolution of these. So salt concentration, as you will hopefully appreciate, looks pretty much diffusive. Okay, so it's diffusing, and then we can also show that the first order, it satisfies the diffusion equation. But let's look at the charge density. The charge density begins to start looking like diffusion, but then pretty quickly, it looks like it's reached a steady state at the transport. So if you're an observer looking at the transporter, you don't see things changing. But of course, it can't be true steady state because you're constantly pumping or fluxing ions, so something must be happening in the far field. But so what I want to emphasize is the dynamics of the charge density are very different from the salt concentration. So the question that I motivated all of this is, how do things spread in plane? So I want to know, how does charge density increase or decrease as I'm walking along the membrane far from the transporter? And to understand the spreading, we can compute the second radial moment. Okay, and if it were a diffusive process, this is what you would see. And what's nice here is that if you do the same thing, calculation for C bar, which is the salt concentration, it is indeed diffusion. Now, what happens when you do this for charge density? You get something that looks like diffusion, but with the diffusion constants that's like 5 orders of magnitude higher. Now, you might say, why 5? Turns out that's a system size. So if you increase the system size, you get higher and higher diffusion constants. And this is the equation's way of telling us that it's not diffusion, something is diverging. And so this was the first telltale sign that there might be some long-range effects involved. So now what I'm doing is I'm walking along the membrane, and as a function of radial distance, I'm going to show you the surface charge. By surface charge, I mean just the charge density at the membrane interface. And I'm also measuring the potential difference between the inside and the outside, and that's the transmembrane potential difference. So this, I'm evolving this for about 1 nanosecond. And what we were surprised to see was there was a power law. So there is a power law decay of 1 over r cubed far away. There's an intermediate region where it decays as 1 over r. And let's not worry about this near field region because it's really close to the transporter. And the same for the membrane potential. So this was the first sort of telltale sign that there are some non-long range effects involved. It's not getting screened like we would normally naively think of Debye-Hückel screening of exponential decay. And so the question was to understand what are the origins of these scaling behaviors, what determines this transition from 1 over r to 1 over r cube, and those transitions will become much clearer when I go beyond 1 nanosecond, and what is the seeming mechanism of this fast in-plane reorganization that seems to not be diffusion. And so we can start by making a very egregious approximation, which is wrong, but it's useful to understand the mechanism. And the approximation that I'm going to make is that as charges move from this ion channel, they're going to stay put at the mouth. So they're not going to travel anywhere. And so all you're doing is that you're building up positive charge here, you're building up negative charge here. And everything else is a uniform dielectric. So obviously it's not true because there are free charges that are allowed free to move everywhere. But the nice thing about this is you end up removing the Nernst and the Planck out of the equations, and you're only left with the Poisson's equation. And so here you have Poisson's equation where you're solving for, under this point charge approximation, the electric potential as a function of the delta functions, and all the time dependence is in this Q of t, where you're just depositing them at some rate. Now this turns out can be solved. You can use open up J.D. Jackson or Kovetz, and there are methods that you can use to solve these. So these are the field lines that come. And what's interesting is you can already see that in this approximation, what we built is a dipole. So we have a positive charge here, negative charge here. And a dipole definitely behaves like a dipole far away. So far away from the transporter, you get this 1 over r cube decay, which is the decay of a dipole. But what this was also surprising is that turns out that there is this intermediate length scale where it behaves like a monopole. Now, normally, if you have a dipole, if you're really close to one charge, you will only see that. So you would expect monopolar behavior. But the main conclusion from this is that the presence of the membrane, because of the fact that it has a finite thickness, and also because it has a low dielectric permittivity, it enhances this monopolar region. So there is an intermediate region, which is of the order of 100 nanometers, where charges on this side just don't see the other side. Okay? And that's an effect of the membrane. And so with this approximation, we get an analytical solution. How well does it do? It does really well until 1 nanosecond. Okay? But beyond 1 nanosecond, it's going to fail. Why is it going to fail? Because there are free charges everywhere. They're allowed to move. And when they move, they will change the potential. And this approximation doesn't take care of any of those things. And so beyond 1 nanosecond, as you will see, is that things are starting this electrostatic approximation is not going to do well when it comes to explaining the numerics. So what's happening beyond 1 nanosecond? And this is where numerics was key to giving us some clues. So what I'm going to do is I'm going to evolve these fields beyond 1 nanosecond. And so here you have this initial monopolar decay, which is 1 over r. And then you have this 1 over r cube. And what you will see we are tracking is the shoulder region from the transition from the monopole to the dipole. And what we saw was that the shoulder region is moving at a constant velocity. Okay, so there is a constant velocity v that we can empirically estimate that it's moving. And so now the questions was, okay, we have this picture where you have an ion channel that's open. It creates this sort of dipole. And what's happening because the membrane is that there is an extended monopolar field, there's an extended dipolar field. But as a function of time, as this ion channel is active, this monopolar field is expanding at a constant velocity. And so the last question was, what is determining the shoulder speed v? And so this is the theory. This is one slide of math, but I will show you some more in a couple of slides, something that's more physical. So the way we solved this was to use analytical transforms. So these are the equations that we are solving. So they're called the Debye-Falkenhagen equations. So this is the equation for the charge density. It looks like a reaction diffusion equation, but it's not a reaction diffusion equation because there's a boundary condition involving potential. And then we are also solving for Gauss's law inside the membrane and in the bulk. And so because we're dealing with cylindrical coordinates, you can use these integral transforms, which are the Hankel-Laplace transforms. You can get a nice, ugly-looking transform solution in the transform space, which of course is difficult to invert. But the key clue came from the numerical simulations where we felt like at large times and large distances, there's a fixed velocity. So we were seeking an inverse in this asymptotic regime. And turns out you can get an inverse, and this inversion to real space gives you a nice solution. And what's nice about the solution is that it's almost the simplest solution you could have written down to explain the data. Because if you see this expression, there is r, there is time, and there's also v. So v times t defines a dynamic length scale, right? And so for r much less than vt, you get back the monopole. And for r much greater than vt, you get the dipole. And in this theory, we also get an expression for v. It's a velocity scale that involves only physical constants. And so there is the diffusivity over the Debye length, which is of the order of 1 meters per second. And then what this is telling us is that the mismatch in the dielectric permittivity between the membrane and the bulk and the water adds a factor, and the thickness also adds another factor, and you get something like 40 meters per second. Right? So this is almost like nearing the conduction velocities that are measured. These are all there in the physical constants. And so how well does this do? It doesn't do really well initially because it's a long time limit, but within a few nanoseconds, you can see that the agreement between the theory and the numerics is exact. And after this is just stuff. Any questions? Okay. All right. So just to summarize, the membrane's response to a single ion channel, that's the theory so far. So the key sort of elements that I want you to take away is that all transmembrane currents, because of their nature, they're breaking electroneutrality. And because they break electron neutrality at the membrane interface, you are going to have long-range fields that are propagating. They're not going to get screened because you are breaking electron neutrality over this 5 nanometer region. And the presence of the membrane enhances this monopolar region. The reason we became excited is because now there is this intermediate region where fields are decaying much more slowly, and it could be the basis for how enhanced cooperativity, because now the voltage is going to be larger than what it would if the membrane were not there. And so finally, I also want to mention that there is this, if I ask how is the voltage increasing at a certain distance from this region, it turns out to be a time scale, and this time scale is called the capacitive time scale. It turns out to be much faster than the bare electrolyte time scale of relaxation in a regular electrolyte found by Bazant and many people before him. And this was published in a recent paper, and interestingly, around the same time, Professor Vlahowska's group also had another paper where they found this time scale. So this seems to be the time scale for membrane charging and discharging, and it's of the order of a few microseconds. Okay, so what I want to spend time the next 10, 15 minutes or so is just to talk about the extensions to this. So now this is a single ion channel in an unbounded domain, and now we've extended this theory now to thinking about what happens when you have electrodes or when you have other bodies, especially confined spaces. What happens when you have geometry? So for example, you have a single ion channel in a spherical vesicle, a single ion channel in a cylindrical axon. And the ultimate goal here is that the single channel theories can now be the input to thinking about how do collections of channels behave on a membrane. And so we are not there yet, but I just want to talk about one of these problems to illustrate a much more general approach to get these solutions that was hard in this initial study where we had to go through these integral transforms. So let me just look at one variant of the problem that I've described. So what I showed you was a membrane with a single ion channel and an unbounded electrolyte. But now you can imagine that there are electrodes here, or there could be other bodies, other cells that are bounding this, introducing another new length scale, L. Okay? And so what does this do? At the very beginning, what these boundaries do is that they change the electric field lines. And so if I look at the dynamics of membrane potential, over here, what's going to happen is that as this channel is active, I'm creating a bilayer here, I'm creating another bilayer here. I'm also going to have induced charges at the electrode interface. And those induced charges are now going to screen everything happening in the bulk, and that's going to change the dynamics. And so here I'm comparing the transmembrane voltage along the distance in both the unbounded case, which I've described earlier, and also where I have electrodes bounding. And you can see that beyond a certain time, the dynamics are qualitatively different. So for R less than this L, it looks pretty much like unbounded for a long time. But then for R greater than L, you start seeing different dynamics. And that eventually also seeps into these are confinement effects playing. So the way we solved this problem was to decompose it, again, because linearity holds. The full problem is where you have an active ion channel and you have electrodes held at a certain voltage. Turns out you can decompose this into a voltage response, which we've solved in a separate paper in Farhadi, and a current response where you have a current, but then you have grounded electrodes. Okay. So that turned out to be the problem that we were solving. And then we initially went through the same approach of using, formulating the equations, using Hankel Laplace transforms, but it was getting unwieldy. And so what ended up being much more useful is a boundary layer theory. So boundary layer theory is sort of, from fluid mechanics, is where the idea is that you have a small parameter, and so you can reduce your equation. So the governing equations as a whole involve these Nernst-Planck equations or Debye-Falkenhagen equations, and you have these boundary conditions. So these are a set of PDEs in R and Z. You have various boundary conditions, and if you have to solve them using integral transforms is for different geometries, different boundary conditions, they quickly become unwieldy. And so how does boundary layer theory work? So here we are exploiting a small parameter. And the small parameter is the fact that the Debye length, lambda d, of 1 nanometer is much smaller than any other relevant length scale. And so that creates a small parameter. And as a consequence, what you get is the bulk, as long as it's in a large length scale, is almost always charge-free. So the Poisson's equation ends up becoming a Laplace's equation. The Debye layers, which are the boundary layers here, end up becoming 1D equations because they're thin. It's a thin film approximation. And finally, what happens in these cases is that your boundary conditions end up becoming time dependent. And what's nice is now you can use all the integral transforms on this set of reduced equations, and then they're much more, I would say, it's easy to obtain solutions for various different geometries, and they also make much more physical sense. And so what happens in this case, I just want to quickly show you the results, is that I'm showing you here how the transmembrane voltage changes as a function of R. So you can see these interesting dynamics. And if I were an observer sitting at a particular location and watching the transmembrane potential rise, this is what I would see. And again, this is several orders of magnitude. It's a single channel, so you don't see large voltages. But even here, what we find are 4 different temporal regimes. So you have this regime below 1 nanosecond where it's almost linear. And then there is this other capacitive time scale where it's sort of sharply rising. And finally, it seems to exhibit saturating behavior. And so the nice thing here is that this boundary layer theory can be used to obtain asymptotic expressions in all these regimes fairly quickly. And so what I'm going to show you here is just the physical picture of how the charging dynamics are changing together with a quick flash of the analytical result. So in this early regime, we are below the Debye time. So things are not diffusing, so the point charge approximation holds. So you can make this electrostatic approximation and get a solution, and that basically just means that there is very little diffusive reorganization of ions anywhere in the system. It's just Poisson's equation. What's happening next is that you have these 2 capacitors. One capacitor is the membrane capacitor, which can hold charge. The other capacitor is the electrode interface, which can also hold charge. In this regime, which we call the capacitive regime, you are charging both of these capacitors, and they're symmetrically charging. So all the ions are flowing to the interface. But what happens then is that these 2 capacitors don't have the same capacitance. The membrane has a much, much lower capacitance because of its low dielectric permittivity. So here, the membrane capacitor is charged, but the electrode capacitor is still charging. And finally, when both capacitors have charged, the charge has no option but to move sort of along and charge other parts of this. And this is what we call radial transport. So again, we can obtain asymptotic solutions for all these things. And the nice thing here is that in this last regime, the diffusive regime, a cable theory-like equation naturally emerges. So we get a PDE that looks very much like cable theory, and it can be interpreted using sort of the cable theory-like ideas. The only difference here is that the length constant, the space constant, these are all like not fitted parameters. They just arise naturally in this geometry that we've assumed. Okay, so I want to sort of end by saying that we can think of all these cases, these responses, in this nice way where we have space and we have time, and the various parameters of the system set up these different length scales and time scales. And each of these length scales and time scales ends up partitioning the space-time diagram into different charging regimes. So in this original case of an unbounded electrolyte, we have this near field and low time. But then the main thing we have is this transition between this monopolar and the dipolar, and that is defined by this sort of dynamic length scale. What happens when you have bounded electrolytes is that the space-time diagram is now perturbed, and there are different charging regimes. And finally, if you say that I don't want blocking electrodes, but I want electrodes where reactions happen, that turns out to perturb the space-time diagram in a different way. So all of this gives us a physical picture of how the activity of an ion channel is charging the membrane, and things are propagating along the membrane. I just want to sort of quickly flash is that now, let's say all of this is for a flat geometry. What if you have a spherical vesicle, and if you have a transporter on the vesicle? So of course, here things become a lot more complicated, cylinder even more so. But the boundary layer theory is helpful, and we can get an expression. So Sirui Ning, the string theorist, sort of got this formula, and it completely exactly describes the behavior. And what this is showing is that within this capacitive time scale, the entire cell feels the effect of the single transporter. And that's the space-time diagram. And for the aficionados, the nice thing about the boundary layer theory is that it also makes the computation much, much easier. So with the boundary layer theory, we no longer need to resolve these steep gradients in space and time. We can use much more coarser meshes, reducing computational times by almost 2 to 3 orders of magnitude in these cases. And you can see that outside of this early time, the boundary layer theory and the full nonlinear finite element completely sit on top of each other over several orders of magnitude. So what's next? We are really interested in now going forward, thinking a little bit about how do multiple channels behave. And this is interesting not just in sort of native biological systems, but also artificial engineered systems. If you can engineer, let's say, a membrane or an ionotronic system with different types of ion channels, what are the emergent dynamics of these channels? Curved geometries, I described spheres. We're also interested in axons. And then the thing that I've not described here is that ultimately the membrane is not a flat, rigid thing. It's capable of flowing, it's capable of moving. And so you may ask, how does a voltage-sensitive channel couple to a mechanosensitive channel? And that's this question of voltage-mechanical coupling that we are also beginning to think about. And so just to conclude, I want to say that ultimately what I've shown is an example of diffuse charge dynamics near membranes as a framework to understand how do signals from ion channels propagate. There is this capacitive timescale that determines pretty much in all cases the leading order charging dynamics. And then boundary layer theory seems to be a generally powerful framework for obtaining asymptotic solutions to these problems. I just want to sort of end with a couple of philosophical notes. So S. Chandrasekhar is a big name in this area. He wrote this amazing set of essays called Truth and Beauty, and where he distinguishes between not just fundamental or applied science, but also basic science versus derived science. And what he says is basic science seeks to analyze the ultimate constitution of matter and the basic concepts of space and time. But derived science, on the other hand, is concerned with the rational ordering of the multifarious aspects of natural phenomena in terms of basic concepts. So what I've shown you is an example of derived science. All the equations have very well known. They've been known for almost 100 years, but their consequences are far from trivial to predict, even in such simple systems. And finally, the other thing that sort of inspires all of this is a famous essay by Phil Anderson, is where just because you know the elements doesn't mean when you put them together, you know the emergent properties. And again, the question that we think about is how do systems of ion channels show interesting and emergent behaviors, and what kind of frameworks are needed to predict them. So with that, I'd like to thank once again my collaborator, Kranthi Mandadapu, who's been a great sort of partner in this set of efforts, and our trainees, Youngju Ro and George Fernandez, whose work I mainly described, and more coming. Thank you.
So I know this is a bit beyond where you are at this point, but seems like an axon with a myelin sheath and nodes is kind of intermediate. It's like the unbounded and then the bounded solution in a cylinder. Are you heading towards there and trying to predict conduction velocities as a function of amount of myelin wrapping and channel density?
So I would say in a naive way, the myelin wrapping, so the myelin adds a thickness, right? And it adds a layer of different dielectric permittivity. And so if I showed you this sort of this particular conduction velocity, you can clearly see here that this basic velocity is enhanced by the membrane thickness, and with the myelin sheet that will only be higher.
Got it.
And even the permittivity, I think myelin's permittivity is probably slightly higher than that of a membrane of lipids. That would change this. So the theory has that, but of course there could be subtle things like if there is free charge between the myelin and the membrane, all those would add complexities. In terms of the question that you mentioned about the nodes of Ranvier, I think that presents some interesting questions from the point of view of coupling between voltage and mechanics, because there are interesting sort of compositions of ion channels there. So we're not thinking about that at the moment, but that's something at least certainly inspires the design of these problems.
We have a question from online. From Petya, for the first problem, if you consider just a 2D instead of a cylindrical geometry, is it easier to invert?
So it is 2D in the sense that, so thank you, Petya. It's 2D in the sense that it's axis symmetric. So I don't know what you meant in terms of whether we're further reducing the dimensionality. Maybe you could just.
There's a follow-on question, maybe was.
Yeah, and then just to be clear, we are solving 2D instead of space and time. So I don't know whether there's further, but I'll be happy to chat when we chat in a few minutes. The second is, I don't quite get the physical effect of propagation. Does it mean that at long times the electric field is described by the monopolar polar field? Yes. So what this theory predicts is that if you keep running an ion channel on a flat membrane forever, ultimately it'll be a monopolar everywhere. But of course, that doesn't happen. So at some point the ion channel shuts off, and then we also describe the relaxation dynamics. And in the relaxation, what happens is that the monopolar dies very quickly, and then finally the dipolar field also dies.
With alluding to what you mentioned about how these membranes have ripples and all these things in biological systems, what do you think the curvature of local curvature around these channels would affect these scaling laws that you found?
So the question is, how does curvature affect these scaling laws, right? So I showed you one example of over here where the sphere, right? So sphere has a curvature, and so imagine that there's an ion channel that sort of starts over here, but the interesting thing compared to the flat is that all the charge has to sort of stay bounded in this closed system. So what's going to happen is that when the ion channel starts, initially things look like, so here, this is the distance, the absolute distance. So at any point of time on the surface, you have an absolute distance d divided by 2r. So initially, things look like it's the dipole and the monopole. Okay, so that's what you're seeing at very initial times. Then what happens is that this monopolar region, just like in the flat, it keeps expanding. So it keeps expanding here. So that's the monopole. And then ultimately, there's no other place to escape, right? So it sort of reaches the end. And what happens is that the long time behavior of this is a uniform potential. So the potential almost becomes uniform. So it's almost like people who do sort of wholesale patch clamp or sort of wholesale recordings, they're measuring a little point, and then they sort of assume that that's the voltage of the entire cell. And what this is just saying is that there is a certain time scale over which that happens, right? But eventually, for a sphere, you have the entire cell, other than the small region, being almost at a uniform voltage that sort of keeps increasing. For a cylinder, now you have both the radial part, which behaves like which is closed, but then you also have the axial part. So there is a combination of those two things playing.
Thank you.
I want to bring it back a little bit, maybe to the biology, since you have sort of a physics treatment of what's happening here. So this works in a context where the cell is as a potassium-sodium pump, trying to establish a potential difference, and then at some point releases that, allows sodium ions through, and so on, and that's the operation that allows the cell to function.
Yes.
So what can you say about that? Is the way neurons mostly do it, is it efficient, for example, in terms of how this pump operates to build, or how the release mechanism operates? Could it be done with different ions? Presumably, yes. So if we think now about what we learned from the physics and take it back to the biology, what would you say?
Okay, so your question is about energetics and how energetically efficient it is. So
perhaps also dynamics, right? Because that's usually involved as well.
Right. So I want to just mention sort of how so what I've shown you here is that the little thing is a pump.
Yeah.
It's taking sodium ions from one side and depositing them on the other side. Now, the issue there is that I've not considered anything. So the way the model is it's a constitutive equation, meaning that I'm just imposing that there is a flux where sodium is being taken from here and being deposited here. The only way it's being constrained here is that there is the energetic sets the time scale over which that can happen. Now, I don't know whether the pump is operating at its maximum possible flux. There are certainly studies of ion channels that tell that say that they're operating at the sort of the maximum Pareto maximum of efficiency versus selectivity. I don't know the answer for the pump, but all the studies that I've shown is what happens after. So I don't have anything in the theory that can comment on whether the pump is operating at its maximal level of efficiency.
Another question from Bob.
Yes.
About, have you considered solving the classical voltage clamp system to connect to existing?
Yeah, Bob. So we have not yet solved the classical voltage clamp system. We're trying to formulate it, but at the moment, initially there was going to be part of this electrode paper where we are setting the electrodes at a certain voltage, but we've not yet solved it.
So what's known about how ion channels like on the same membrane couple to each other? And I guess like based on like physiological structure of how they're organized, do you think that they would act, cooperatively or mostly independently?
The answer is we don't know. The few studies, the super resolution studies that exist out there, says that the way ion channels are arranged relatively to each other, especially in the nodes of Ranvier, are not random. So there's almost like a periodic spacing of one type of ion channel and a different periodic spacing of another type of ion channel. They sort of and all of these are, the spacings can be of the order of 100 nanometers or even slightly lower. And so given the time scale that we find, there's obviously the exciting possibility that there could be coupling between them. So one is kind of voltage coupling. So which is that if I have, let's say, one voltage gated ion channel opening, could it cooperatively induce or bias the opening of a nearby voltage gated ion channel? That's one kind of question that we are after. The other, of course, is mechanical coupling, right? So can increase voltage here change membrane tension or moduli in its neighborhood? Again, I don't know the answers to these questions, but there is a hypothesis that those things may be present. And there have been studies that have used still macroscopic current and voltage experiments to sort of deduce whether these things exist, but I would say there's no microscopic theory.
I mean, related to that, they're on the same scaffold, even, right?
Yes.
I mean, they don't even need to communicate through the membrane. They could communicate through the scaffold that's even holding them mechanically.
Right, but
to curve the membrane, like they're all on protein complexes.
So you mean the
nodes, at least.
no. So you mean the cytoskeleton?
Yeah, kind of.
Yeah. The question of whether it's the cytoskeleton that allows that coupling or it's the membrane, I think it could be the membrane also, because Thank you.